Okay, we're going to finish up chapter 9 today. I've know I've been slow getting this out. Couple reasons for that. Seems like in this section here, 9.4, they just throw a lot of stuff at us. Uh, so this will be a little bit different lecture. I'm going to just try to hit the high points. There'll be lots of details in here that I'm just going to skip over, and you can take that as a hint that that won't be important. It's amazing how much information they packed into this little section, and I'm not sure it was all fleshed out, as well as we're approaching the end. So let's get the high points out of the way and give you some things that you should understand about membranes. And the first one is that membranes are fluid. And so they've done some really interesting experiments to show that membranes are fluid. And one of them is this labeling of proteins with different fluorescent dyes, as you can see here. And so you can see that they labeled the mouse cell with a dye that fluoresces green and a human cell with a dye that fluoresces red. And then they use this Sendai virus that will actually bind to both cells and actually cause them to fuse. And so when it first fuses, you can see this picture down here where you can see the difference between the location of the green and the red fluorescent dyes. Then they just leave it for 40 minutes at body temperature, 37 degrees Celsius, and the red and the green markers have fully intermixed. Now, what they did is they use a special filter so you can only see the green dyes and the red dyes. And that's the same here. This is, I believe, a composite image of a picture taken where you only see the green and where you only see the red. And so after time, they mix, which lets you know that those proteins that are labeled are actually moving throughout the membrane there. And so this is the fluid mosaic model. So instead of everything being fixed in the bilayer, things are able to move. And so one way to think about that is that these integral membrane proteins, those that are buried within the membrane, are like icebergs that are floating in a two-dimensional lipid sea. Sounds poetic. So they're able to diffuse laterally unless they are attached to some interior element. So in some cases, they don't move or they're restricted, and we'll talk a little bit about that later. But from that previous experiment, we can see that they do have the ability to move. And so, yeah, in this fluid mosaic model, you can see those little gold things are representing cholesterol. You can see different types of transmembrane proteins. You can see these that are just presenting on one surface. You can see those that are attached by the lipid groups. So we've talked about all these, and then you can see that the proteins have carbohydrates on them. So membranes are much more, at least biological membranes, are a lot more diverse, and I would almost say complex than just a lipid bilayer. There's a lot going on, and that's kind of what we want to understand here. So we talked about how the lipids can actually move translationally, meaning through the same leaf of the bilayer, but they tend not to flip-flop. And we'll address those topics. But again, an interesting experiment to show that these membranes can actually move is, in this case, they have bleached the, they've attached a fluorescent probe to a protein, and they've also done this with lipids. So this experiment, which is known as FRAP, which is fluorescence recovery after photobleaching. The experiment could be used to both show that lipids move or also proteins. So essentially what you do, it's like the first slide, you label something, like I said, either lipids or proteins in there. And then you take a picture, use a fluorescence microscope, and you can see that that dye is evenly dispersed over the entirety of the cell. And then what you do is use a laser and put a lot of power in, and fluorescent probes tend to bleach. And what that means is they just lose their ability to fluoresce. And so you can shine this on a spot, and what you see is here's the fluorescence intensity. You bleach it just by hitting it with a, not necessarily a high-powered laser, but one that will cause the fluorophores to no longer fluoresce. And then you can see that there is a recovery time. And that recovery time is as the molecules, again, either the membranes or the lipids, re-enter that bleached spot over time. And eventually you'll see that you get recovery. You're not going to get up to 100% recovery because you've destroyed some of the fluorophores, but essentially you get near 100%. It really depends on how much of the surface you bleached. But again, what this shows is that the membranes are actually fluid. Things are moving within the membrane. So again, this is known as FRAP, fluorescence recovery after photobleaching. So if you just specifically label membrane proteins, what they find out is that 20 to 30% of the membrane proteins are freely mobile. So they diffuse an order of magnitude more slowly than lipids. So they're not as quick in their movement. And even some proteins move more slowly. And then essentially some are going to be immobile. So this is just a general example, but if you labeled a specific protein in here and you only saw partial recovery, or you saw a very delayed time, or let's say that you never saw recovery, that would give you information about the mobility of that type of protein. So like I said here, you can use this experiment to look at the mobility of lipids or of proteins. So like I said, if you wanted to label a specific protein and it was found dispersed throughout the entire membrane, and then you photo bleached, you could see how fast it moves or even if it does move. Like I said, this FRAP experiment can tell you the mobility of proteins as well as lipids. But again, the take-home message is that things can move within the membrane. And again, some proteins are going to move better than others, and we'll talk about that, what holds them in place. So we know that there's proteins inside of cells that actually give them structure, and this comes from what are called erythrocyte ghosts. So what you do is essentially you osmotically lyse the cell, put them in very pure water, and then the water is going to move into the cell and cause it to burst, and all the stuff inside the cell actually leaves. And then as you clean that out and get them back to a regular physiological conditions, that membrane will actually reseal, and the erythrocytes will take on their original shape, but they tend to be translucent because they no longer have the heme inside of them. And so something that they've done in this is tried to understand their properties and why erythrocytes are good at delivering oxygen and able to travel through cells. So if you actually took a slurry of solid particles that are about the size of red blood cells, and meaning they're in water or a medium similar to blood, the red blood cell, or sorry, yeah, that medium would flow about as well as sand. Meaning there's something special about these erythrocytes that allow them to flow through our blood much better than sand in blood. So it has to do with the shape and their ability to flex. So like we mentioned, there is a cell has a skeleton that stays inside the membrane. And so this membrane actually gives the cell shape. Now they're not tightly attached to each other, which allows these molecules to flex. And so instead of being solid or rigid bodies like sand, they tend to be more fluid-like and easily deformable, so they actually flow much better. So to connect to the previous thought, if these were rigid particles, and you'd find that they are much more viscous or have a much harder time flowing. But we do know that they are flexible and they tend to be more fluid-like. So even though they have a skeleton inside, that skeleton is flexible. So one of those things that actually makes up the skeleton is the spectrin protein. Now, I spent way too much time trying to figure out the book. I think they, let's just put it this way, did a poor job of explaining this. And so I'm just going to leave it at that the erythrocyte skeletons are made mainly of spectrin. I had to go back to the original paper to try to figure out what this drawing even means out of there. For some reason, this was really bothering me. So we're not going to really talk about the structure of spectrins. I don't believe the book, like I said, gives an adequate description nor a good image of how these spectrin proteins associate. And that's the same with the ankyrin structures. So like I said, this lecture will be a little bit different. So definitely know that spectrin is used in creating that skeleton and ankyrin as well. But the take-home message we can actually get is from this picture right here. So here's the erythrocyte skeleton. And so off to the left is an electron micrograph. And so you can see that there's these structures here, and they kind of come to a common point there, and they're interconnected. And then off to the right, we have a cartoon structure. And so let's make sure that we understand what's being represented here. So they're folding down this panel. So anything on the top would actually be in the cytoplasm or the interior of the cell, and anything on the bottom would be on the outside. So they're actually showing you the interior of the cell. And so, like I said, we got these spectrin proteins here. You've got these ankyrin proteins here. And so what I want to stress is that there's a big network of interconnectivity on the interior of the membrane that adds some rigidity to the protein, and also will anchor some proteins in specific spots. So when I talked about not all proteins have the ability to move, this is why some of them are actually anchored to this cytoskeleton here. And there's a lot more parts to this cytoskeleton that the book doesn't talk about. So again, for shortness of the time, and I think some deficiencies in their explanation, we'll leave the cytoskeleton take-home message as there's lots of proteins that are interconnected that form a meshwork that anchor proteins, cross-link proteins, and that is where the cytoskeleton comes from. And so within the cytoskeleton, we can actually start to come up with a model of how things can move or have restricted mobility. So this takes us back to why some proteins are freely able to move and some are not. And so this green is just kind of a schematic representation of all those interconnected proteins. In fact, you can see that it kind of matches up this picture over here. And so if we want to understand how proteins are mobile, you can use this gate and fence model. So if we look at A there, it's going to interact tightly with the membrane skeleton. And so we would expect that type of protein not to be mobile in the membrane. And B, let's say that it doesn't interact, but it's caught in one of these little traps here. So it's sticking out above the membrane here, and it's got a portion buried in the membrane, but it really can't jump over that skeleton there. And so it would be able to have some localized movement within that region. And then there's other proteins that are mobile. And so either the skeleton isn't completely formed, or it's maybe just buried in kind of the upper leaflet here. It can sneak under some of these. I don't believe that they're going to jump out. I'll say that's not going to happen, but there are proteins that can find the holes in the cytoskeleton and be able to diffuse freely, or they're just not, their interaction with the membrane is such that they're not going to be inhibited in their movement by the cytoskeleton. Okay. So that's what we'll leave with the cytoskeleton, is that there's a lot of interconnected proteins that offer structure to the cell, as well as places for proteins to be anchored. Now, something that's really interesting is that membrane lipids are distributed asymmetrically within the bilayer. This is something I've been fascinated with since graduate school. And how they've studied this is you introduce a phospholipase, and remember, those are enzymes that will release free fatty acids from the lipids. Now, they tend not to go across the membrane, and so what you can do is have it cleave off the lipids on the outside, and you analyze those. And there's other methods to do this, but that's one of them. And so you can see the total distribution of phospholipids. Well, that should make sense in that you have 50% on the inside and 50% on the outside. But of those phospholipids, we see that sphingomyelin tend to be found more on the outer leaflet, as well as phosphatidylcholine. Phosphatidylethanolamine tends to be more on the inside. And interestingly is the phosphatidylserine is almost exclusively on the inside. And so cells that actually change this distribution of lipids, that's often a signal that they need to be retired from service. And so there are enzymes, and we'll talk a little bit about them in just a second, and a little bit more in the next chapter, that are called flipases, that actually help maintain this lipid asymmetry. If you lose this lipid asymmetry, that is a signal of either a disease state or a signal that that cell needs to be retired. Helps it undergo apoptosis, which is programmed cell death. But I find it fascinating that our cell membranes actually have this order in there, this asymmetry, and if we get rid of that asymmetry, that is bad. Okay. So there's other places in just this, instead of the cell membrane, where we find lipids and membranes. So in eukaryotes, membrane lipids occur in the endoplasmic reticulum membrane, and that is contiguous with the nuclear membrane. So there's actually lots of different membranes in there. In fact, if you remember back to the symbiotic theory, it was thought that a lot of the organelles that occur in cells came from symbiotic bacteria living inside other cells. And so in the mitochondria, you actually have double membranes in there. You got the Golgi apparatus that has membranes. You've got the endoplasmic reticulum and the rough endoplasmic reticulum and the nuclear membrane and lysosomes. So we don't just use these membranes to surround the cell, we also use it to exclude different areas within the cell or to compartmentalize within the cell, and that offers a lot of possibility for regulation. And so one way that we can try to figure out how this lipid asymmetry arises is they did an experiment in bacteria. And so they grew the bacteria with a short pulse of a radioactive phosphate. So you got P32, which is a radioactive form of phosphorus. And so what they did is they gave a little bit of this phosphate, and that phosphate actually is pulled into the cells and incorporated into the phospholipids. And then they use this trinitrobenzene sulfonic acid, the acronym TNBS. And what it does is it reacts specifically with the phosphatidylethanolamine there. And you can label the outer membrane with that. And then what you do is you use that tag, the TNBS tag there, and you pull out the lipids that specifically have that tag. Okay, so let's see how that works schematically here. Okay, so we can see in this diagram here that the TNBS, well, sorry, let's walk through it. So we start with and this is going to represent a bacteria here. So give it a little bit of phosphate. That phosphate is brought into the interior. The bacteria are going to start to synthesize these phospholipids. And if we, right after we introduce that phosphate, we use the TNBS, we're going to label the proteins on just the outside. The TNBS isn't going to get into the inside. And then what you do is you separate those labeled lipids, and what you find out is none of them are radioactive. And if you do another experiment, again, incubate it for a short amount of time with the radioactive phosphate, and you wait just 3 minutes, and then you go and label it with the TNBS. Again, you're just going to label those on the outside, and then if you separate, you'll find out that you have some radioactive labeled phospholipids. And so what that lets you know is that the synthesis of these phospholipids is occurring in the interior, and that there's some method for them to move out. If you remember that this transverse movement is actually really, really slow in membranes, because that polar head group has to bury itself within the interior and move to the outside. So this is what's referred to as flipping or flopping. And so the flip-flop rate for phosphatidylethanolamine is 10 to the 5-fold faster in bacterial membranes than in synthetic lipids only. So if you just had this that was synthetic and we set it up, as we talked about before, the movement from of a lipid from the outside in or inside out is very, very slow. So something is helping those lipids move from the interior leaflet to the outside leaflet. So a really interesting experiment to show that something is in control of establishing this asymmetry in the bilayers. And so they're thought to be what are called flipases, so membrane proteins that facilitate the flip and the flop of specific phospholipids. And so I probably shouldn't say equalize, but what they do is they establish the distribution between the inner and outer leaflets of the membrane. And they do this through facilitated diffusion, which we'll talk about in the next chapter. And then they got the phospholipid translocases. And so these are going to be ATP dependent, and they're going to transport a specific phospholipid. And these are going to create, oh, so actually, sorry, I need to go back. Since these are facilitated diffusion, I apologize, these are going to equalize the distribution between the inner and outer leaflets of the membrane. So you're not going to have much control. This is, again, facilitated diffusion. So you're going to set up an equal arrangement of those lipids. If we need to set up an asymmetric one, then I have to use active transport, and this is going to create the unequal distribution of lipids between the inner and outer leaflets. And again, we'll talk about this in chapter 10. So take-home message is the distribution of lipids biologically important, and so if you remember, phosphatidylserine is almost exclusively found on the interior. If you move it to the exterior of cells, especially on red blood cells, that indicates that they need to be removed from circulation, and it also induces blood clotting. So let me just go back to that table, and so you can see, yeah, phosphatidylserine isn't typically found in the outer leaflet. So if you start to see it found there, that is a signal to remove that red blood cell, or it is involved in clotting. So this arrangement of lipids here, like I said, there's enzymes that do that to maintain the asymmetry in the membrane. But we do know that the phospholipids are synthesized on the interior, and they get to the outside of the cell much faster than just by diffusion by themselves. So that diffusion can be facilitated by flipases, or it can be actively transported by phospholipid translocases. So something else that we need to talk about is how do proteins actually get into the membranes? And so in eukaryote, membrane proteins are made in the rough ER, the rough endoplasmic reticulum. And so if you remember why they refer to it as the rough endoplasmic reticulum, that is because the ribosomes are attached into the membrane. And so, again, the synthesis of lipids is occurring in the cells and the proteins, and so somehow they need to get to the cell membrane. And so membrane lipids and proteins are packed into vesicles that bud off of the endoplasmic reticulum, and then they go and fuse with the Golgi membrane. And then in the Golgi membrane, they bud off and refuse with the Golgi membrane, and then bud off and refuse, and eventually make their way to the plasma membrane. We'll see that a little bit later. But what we want to do is talk about, well, another reason that we have asymmetry in the bilayers. So not only is there asymmetry in the bilayer, there's also what are referred to as domains within the membranes. And so epithelial cells have an apical domain facing the lumen, and these often have special functions. And then the basolateral domain covering the remainder of the cells. And so again, the actual bilayer here may have differences in the type of lipids, but also within a cell, there may be different functions here than there are here. So if you look at the overall cell, there may be different parts. So let's look at those. So you get these microdomains within an actual cell, and they may be enriched in a particular lipid and or protein. So these particular lipids may be clustered around an integral protein. So sometimes you see a protein that's embedded in the membrane, and it tends to be surrounded by a specific type of lipid. Some of these lipids will interact with metal ions like calcium-2, and so they'll cluster. If you expose it to calcium, all of a sudden you start to cluster a specific type of membrane. So what that means is not only is there asymmetry in the bilayer, what that means is there's regions within the bilayer that are heterogeneous from other regions in the bilayer. And so this leads to this idea of a lipid raft, so a particular type of microdomain. And so this is going to be closely packed glycosphingolipids, so like the one that's showing off to the right there, and cholesterol. It'll prevent the bulky head groups. Well, sorry, the bulky head groups will prevent the tails from tightly packing. And then they'll allow the cholesterol to sneak in between those unpacked tails. And what that does is it creates an environment that some proteins like to preferentially interact with. So you'll get these rafts of specific types of lipids in specific arrangements, and that will attract or be more favorable for certain types of lipids. Okay, but let's talk about how the proteins actually get into these membranes. So there are ribosomes in the cytoplasm, and if we tried to make a protein in the cytoplasm and that protein folded, it would be too hard to transport across the membrane. So especially if it was a secretory protein that you needed to get outside the cell and deliver elsewhere. So in eukaryotes, most soluble and mitochondrial proteins are made by free ribosomes. But there are ribosomes that are bound to the endoplasmic reticulum, which is a membrane, and they synthesize these integral membrane proteins, so those that are stuck in there. And they also synthesize those that are found in vesicles and those that are secreted, which are about 40% of proteins. So we'll see why. So like I said, just those cytosolic free ribosomes aren't going to synthesize the secreted proteins because they're just not going to transport themselves across the membrane. So the secretory pathway interacts with the ribosomes at the endoplasmic reticulum, and it sends the proteins into the membrane or the endoplasmic reticulum as they're made. Okay, so let's look at step 4. So once the proteins are sorted, how do they get into the ER to be packaged into vesicles? And we can see that in this diagram here. We're going to read it from left to right across the bottom. And so we've got the mRNA that is interacting with the ribosome. The protein is coming out here, and we have a signaling peptide. And so what happens is you get the SRP, which is the signal recognition particle, and it will bind GTP, which is similar to ATP, but you're using guanine instead of adenosine. And it will bind to that signal peptide as well as the ribosome. And then that will cause the ribosome to dock, and you got this SRP receptor, so the signal recognition particle receptor that also has a GTP bound, and that recognizes the signal recognition particle. And then what happens is that signal peptide is put into the translocon. Now, if you notice, this is the lumen, or the interior of the rough endoplasmic reticulum. So now what you've got is the ribosome associated with the membrane of the endoplasmic reticulum, and you've got a protein that's being fed in there. Now that signaling peptide, which is what indicates that this needs to be dropped into the endoplasmic reticulum, is cleaved off. And then as the protein is made, there is some processing. You can add carbohydrates on there. And then either this this protein is released into the endoplasmic reticulum for further processing, or it is incorporated and embedded in the endoplasmic reticulum right there. So note in this pathway, folding of the integral membrane proteins into the membrane is also done by a ribosome translocon complex. So like I said, if this is going to be an integral membrane protein, as it's coming out of the ribosome, it is packaged into the membrane as well. Prokaryotes use a similar pathway to make their extracellular and integral membrane proteins. So again, the thing is, how do we get proteins outside of the cell and into the membrane? And cells have other mechanisms to get the proteins out of cells or into compartments. And there is 2 different examples, but that will be for your own interest to read. But let's talk about this signaling sequence. These are found on all secreted endoplasmic reticulum resident and lysosomal proteins, as well as on many integral membrane proteins. So again, these are going to be the ones that need to be stuck into a membrane or released from the cell or packaged within a membrane. These aren't going to be the free cytosolic proteins. And the signal sequence is at the N-terminus, and that makes sense because we synthesize proteins from the N-terminus to the C-terminus. And so it's released from the ribosome as the translation starts. So it's going to be one of the first things that comes out of the ribosome. And so here you can see some of that signaling sequence. And so you can see that they consist of a lot of hydrophobic residues. Now there's one or more basic residues in there, but you get a hydrophobic sequence that can form an alpha helix, is the one type of these signaling sequences there. Now, again, these translocons are what moves the protein across the membrane. And so these translocons, we need to look at a little bit more. And they tend to be found in all domains of life. So all domains of life have these proteins embedded in the membrane that allow other proteins to be transported across the membrane. Okay, so these translocons, they allow the proteins to be synthesized in the endoplasmic reticulum. They're found in all domains of life. There's 3 subunits, and the key is that they make this aqueous pore that spans the ER. So if you look here, this is the hydrophobic region of the bilayer. Here's where the polar head groups would be. And it spans the bilayer and provides a hole for the protein to move through. And so once we get these proteins into the endoplasmic reticulum, so let's kind of walk through here. So the mRNA is going to come out of the nucleus. It's going to bind to one of these ribosomes, go through that process. And as the ribosome processes the mRNA, the protein's going to, let's pick this guy right here. The protein's going to go into the endoplasmic reticulum. And it's going to get processed as it comes out. Like I said, some of these proteins actually get embedded into the membrane. Some of them are just going to be released and be trapped inside this. And then what happens is you get budding off of the membrane from the rough endoplasmic reticulum. And so it's a little hard to see, but you'll have some integral membrane proteins, and then you'll just have some. These are going to be the ones that are going to be released, the secreted proteins. Well, this then goes to the Golgi apparatus and binds to one of the leaflets there. And essentially through the Golgi apparatus, you get processed and you get carbohydrates added and trimmed and other modifications to the protein. And then eventually from the Golgi apparatus, you'll get these secretory vesicles that then fuse to the membrane and are released. Or the other thing that we need to solve is, well, how do they get into the membrane of the cell? And so how they get into the membrane of the cell is, it's a little bit hard to see, but we've got some that are embedded in the membrane of the vesicles. So when they fuse, they will stay embedded in the cell membrane. And then there's others, enzymes that are just packed into the lysosomes, sorry. And so this is the process to handle proteins, all three types that we were talking about, those that are secreted, those that need to get into the membrane, and those that are packaged into lysosomes. Now, those proteins that are just floating around in the cytoplasm, those can just be made by just the regular free ribosomes in there. But if you're going to be secreted, or you're going to be found in the membrane, or you're going to be packed into a lysosome, this is the process that you need to go through. Okay, so here is just a little bit closer view. So let's say that it went through all that process, came through the endoplasmic reticulum, went through the Golgi apparatus and got processed with carbohydrates on it. Like I said, as it came through the Golgi apparatus and the translocon, it likely got embedded in the membrane there. And so it's released from the Golgi apparatus in this vesicle, and we need to now embed it in the cell membrane. And so what's going to happen is this vesicle will now come and fuse with the membrane there. And you can see as it fuses, what's nice is those membranes are embedded in the vesicle, are now embedded and have directionality in the cell membrane. Now there's three types of these coated vesicles that go and bind with the cell membrane. They're the clathrin, which transport the transmembrane and the GPI-linked proteins, as well as the secreted proteins. There's a COP1 protein and the COP2. So the best one that's understood is the clathrin. And again, this is one area that we're just going to kind of say, okay, that's cool. So clathrin-coated vesicles, they're the better characterized ones. And what's interesting about them is it's not just a membrane. They're coated with these proteins, which are known as the triskelions, which sounds like something out of a Marvel comic. For some reason, I don't know if there was something called triskelions in there, but they're proteins. We're not going to need to know much about them other than that they coat these vesicles that go and transport proteins to the cell membrane. And in fact, here's a picture of them, and you can see them. They got kind of this tri-armed structure. So if we look a little bit closer at them, don't worry about their anatomy. Just understand that this tri-arm structure you can actually arrange into a couple different things to form hexagons and pentagons. And so you can get different size of these coated vesicles. So these triskelions, they form the edges of these polyhedral cages. And then these clathrin-coated vesicles also participate in endocytosis when we bring things into the cell. So just understand that it's not just a membrane that is going to bind with the cell membrane to transport these, but we have these coated vesicles. And so let's take an actual picture, or a picture of this. So you got the cytoplasm here. You got a specific substance binding to the receptor proteins right here. So we're packaging in it. This is actually going to be a picture of endocytosis. So it'd be kind of the reverse process if we were excreting these outside the cell. But since this is a cytoplasm, we're bringing it into the cell. So We're going to bind to these surface receptors. We're going to, what they refer to as invaginate, pull that membrane inside and eventually close it off. And then you have the clathrin coat. So if this was the interior of the cytoplasm, the same process would happen. This clathrin coated vesicle would bind to the membrane and start to fuse. You would open up, and then you would release those proteins into the exterior space of the cell, or if you had embedded proteins, now they're stuck in the cell membrane there. Okay, so these proteins actually mediate vesicle fusion. So how do vesicles bind to the cell membrane? So we talked a little bit about how they bind to the membrane to embed proteins, but we need to look a little bit more at how vesicles can actually bind. This is a very important process in neurotransmission. We'll talk a little bit about that in cell signaling. But one of the common examples of this is in nerve transmission. And what we have are these synapses. And you've got the presynaptic and the postsynaptic membrane. And then you got the synaptic cleft. You've actually got a gap in between the cells there in nerves. And so they don't directly connect with each other. And so what happens is you actually will package nerve transmitters into a vesicle. So this is a little bit different because we're not packaging proteins, but the same type of idea is that we can package material inside of a lipid-bound membrane. And that lipid-bound, or that lipid membrane can bind to the cell membrane. And when it does, it will release its contents outside of the cell. And in this case, it's a neurotransmitter that then can travel past or through the synaptic cleft and bind to the postsynaptic membrane. And when they bind to the postsynaptic membrane, they induce a change within this cell. And like I said, we'll talk about this process a little bit in cell signaling, but we just want to understand actually how do vesicles go and fuse. As you'd expect, the surface of this vesicle tends to have a lot of negative charges from the phospholipid bilayer. And this membrane here has a lot of negative charges. So in fact, membranes should actually repel membranes, and there shouldn't be a lot of natural attraction for them. And so, lo and behold, this process is mediated by proteins called snare proteins. And there's two types that we'll talk about, the R snares. Now, the R actually stands for arginine. And there's Q snares, and Q stands for the glutamine. So the R snares are associated with the vesicle membranes. And so you can see that in the diagram down here. We've got these R snares, and the Q snares tend to be associated with the target membrane. And so, like I said, these membranes should have natural repulsion for each other because they're both negatively charged. So there needs to be something that will help them mediate their natural repulsion, and that's these proteins. And so as the vesicle starts to approach the membrane, these snare proteins will interact with each other and actually start to form a helice around each other. And as you tighten that helice, that will bring the proteins into close contact, or sorry, the lipid membranes into close contact. And as they start to become into close contact, you get reorganization of that bilayer. And so you can imagine at this point right there, that's some unfavorable interaction. And so you kind of drive these polar head groups away from each other. And so you start opening up kind of a cavity here. Well, that's not going to be necessarily just a vacuum there. What's going to start happening is this hydrophobic region and that hydrophobic region are going to start to move towards each other. So you just get rearrangement of those lipids. A lot of it's driven by the polar head groups trying to get away from each other. And then the advantage of the hydrophobic groups interacting with the hydrophobic groups. And so those snare proteins tend to kind of bring these into close proximity. You start excluding some of those lipids out, and essentially you just reestablish a new bilayer there. And then eventually it will open up and you can release the contents of the vesicle into the interior of the cell, or you can release it to the exterior of the cell. Okay, so that is snare-mediated vesicle fusion. Now, if you ever wonder how you got the flu, similar thing, or how you get the coronavirus, it's a very similar type of idea. So viruses tend to be encased in lipid membranes or sometimes just protein coats. But somehow we need to get that protein coat to get inside the cell so that the virus can be replicated. So there needs to be some type of attraction between there. So influenza has the hematoglutinin protein. And if you're wondering where that name comes from, it actually causes glutination, which is aggregation of red blood cells. So heme from the red blood cells, and agglutination, which is aggregation. And so this hematoglutinin is one of the major proteins expressed on the surface of viruses. And it has and it recognizes sialic acid, or what's called neurominic acid, on the surface of cells, and it'll bind to those cells and actually cause it to fuse, and we bring those viruses into our cells, and this will either have DNA or RNA in it, and then it takes advantage of our cellular machinery to replicate the virus. And as it replicates the virus, that causes more virus, and then what will happen is the virus will assemble at the cell surface, and that's what you see by the term assembly there. It will assemble all those proteins, and it will actually bud off of the cell, and then it's released. And so you're actually releasing more virus into your body, which in the case of COVID will find itself into your fluids, and then as you cough or sneeze or spit, you can transmit that virus to other people, and the same process will occur where that virus will then go bind cell membranes, it's brought into the cells, and then our cellular machinery will replicate the virus, and that person would then have COVID, and this cycle can repeat. So that is why social distancing and masks are being employed to limit COVID. Like I said, COVID is transmitted through fluids. So the mask actually traps the fluids as you're speaking. Your spittle isn't transported to other people. And social distancing, you're not being exposed to other people's spittle, and so those viruses can't get inside of you and get replicated. And to close off, here's some pretty pictures of hemagglutinin. And the key to help it facilitate fusion is a structural change. So I know that was kind of the surface. Like I said, there's lots of different stuff in this. So let's look at this checkpoint 9.4. You should be able to describe the fluid mosaic model. And you should be able to describe how the membrane skeleton influences membrane distribution, why some are more mobile than others. We talked a lot about why biological membranes are asymmetrical, both at the membrane level and also a little bit about the cell level. So what do flipases and phospholipid translocases do? And like I said, biological membranes are asymmetrical at the cellular level, which is what we talked about with the lipid raft. In general, just understand the secretory pathway, how things actually get embedded in the membrane. Include, generally the function of the translocon, how it actually gets the protein from the cytoplasm into the endoplasmic reticulum, and how that helps facilitate getting integral membrane proteins into a membrane. Yeah, we did talk about how a membrane protein and a secreted protein and the lysosomal protein are transported from the rough endoplasmic reticulum to their final destination. We didn't talk about how it makes sense that carbohydrate groups serve as intercellular addressing signals, so don't worry about that. And in general, describe how SNARE works. Why do we need these proteins to help mediate fusion of vesicles or membranes with other membranes? And that should be it for this chapter, and we'll move on to chapter 10.